want to welcome everyone to this product theater. We're going to talk about TissueCypher, and specifically TissueCypher-based risk stratification for Barrett's esophagus. And this is a process which I'll get into, which has defined what we call a clinical practice model. One of the things, as many companies and many devices and many new innovative processes try to seek adoption in clinical guidelines and best practices, is that we as practicing gastroenterologists still need to drive the adoption and utilization of innovation. This is a process that we sought to create to really help drive innovation that deserves to be in the hands of our physicians and benefit our patients day to day. It's really the idea of having a playbook on how to effectively implement innovation in clinical practice. First, I want to thank our team here. I'm going to have them each introduce themselves. I'm Komanduri. I'm a gastroenterologist at Northwestern University in Chicago, and one of the members of this team of key opinion leaders who help create this clinical practice model. So why do we need this? There's so much novel innovation coming in terms of not only devices and drugs and everything else, but specifically in the area of esophageal disease and Barrett's esophagus, as we continue to try to identify ways to reduce the mortality and also the incidence of esophageal adenocarcinoma. And we know we have many gaps. And so we're looking for technologies that can help us identify more patients, help us with risk stratification, and drive patients towards the proper management. And so that's the idea behind this clinical practice model, and that's what the model does. It really helps you understand, how can I effectively do this despite having X barrier, or this in my practice that is challenging to overcome. And then ultimately we educated our peers and we brought on other practitioners to really start to implement the clinical practice model, which extends into today. So with that, I want to get right into this and start talking about what we're here for today in risk stratification in Barrett's esophagus. Thank you.
Thanks, Dr. Komanduri. I'm Dr. Vivek Kaul, the University of Rochester Medical Center in Rochester, New York. So I'm going to go into the second section of this CPM, or clinical practice model, if you will, and speak a little bit to how the CPM concept that Sri just explained was put to play, pun intended, as a playbook for TissueCypher. So in this particular case, we set out with a set of objectives to get some background intel on this. So we looked at the current use of TissueCypher in clinical practice. The idea was to create a roadmap and to create a model that would work for most, if not for all. So with that in mind, the agenda was set, and we thought that a model that is being developed with the use, experience, expertise of folks who've been in the business for a long period of time and seen pretty much a diverse population of patients. It will be good to start with what the current practice is, look at the literature, and then kind of get knowledge from each other as to what works, what doesn't work, how did a particular institution, navigate their challenges, what challenges have not yet been navigated, what questions remain. With that in place, we figured out some of the current issues and then possible solutions, and then also an eye towards the future agenda of where not only this space of predictive analytics might be going in clinical practice, but where specifically TissueCypher will be positioned. Standard risk stratification really heavily relies on endoscopic findings, but also on histopathology, which has its own Achilles heels in terms of the interpathology variability and so forth, and the literature is rife with that. So that's where we are right now. And what does TissueCypher add to that? So with the molecular prognostication using a proprietary set of molecular markers, the idea here is to look at, differentiate the truly high-risk individuals, and some might say even the high and intermediate risk combined, versus the truly low-risk individuals, and add that information, that intelligence, that knowledge to the existing paradigms that are in play today based on our current guidelines and practice. So not necessarily a replacement for histopathology, but an adjunct. So this is not a slide I'm going to go too deep into, but this is a proof of principle to show where the gaps are, even for experts, even on this stage. Here's a 50-year-old healthy CEO, the point being that this is a resourceful patient who has a 6 centimeter non-dysplastic Barrett segment. That's a pretty sizable segment. And this gentleman doesn't want to consider surveillance. He wants to be treated. And the guidelines say don't treat him or her for that matter. So this is somebody where you could sit them down and say, listen, you want to go the extra mile, you want to add another layer of intelligence to your decision making, you could be a good candidate. If this individual came back as a high-risk individual, you're going to be ending up treating this person, and for good reason, right? So that's a good example there. Now, at the bottom, there's an 83-year-old, kind of a different scenario here. He tells you that both of his parents lived up to nearly 100 years of age, and he has a 3 centimeter segment, which is low grade, and the guidelines would tell you could watch him for another 19 years and navigate his osteophytes every endoscopy session. Or you could say, listen, we could do this test and figure out if you're truly low risk and just ride into the sunset. And at that point, you can live as long as you want, but you're not likely to get esophageal cancer in this lifetime. So a couple of use scenarios that positioned the team well, that whetted their appetite, because, people, with a lot of experience and expertise, it's hard to get them interested in a conversation. So with that, I'm going to turn this over to Dr. Harshit Khara, who was involved with the earlier studies, and we'll go through some of the literature on this topic.
So I'm Harshit Khara. I'm one of the interventional endoscopists with a personal passion for Barrett's esophagus, based out of Geisinger Medical Center in Central Pennsylvania, where I serve as the director of endoscopy and the system director of advanced endoscopy. And not only follow these patients for the risk stratification and diagnosis of Barrett's, but also for the management and the treatment of Barrett's, so the entire spectrum. So this definitely is an integral part of our practice. And as Vivek mentioned, we've been part of the original pivotal trials. So we've been part of the science behind this. And I'd love to share that with all of us, that it's not just a commercial test, but it definitely has strong roots of where it developed and great branches of where it's growing into. So I'll go over the GAP1 study. This was the initial validation study comparing non-progressors to progressors. So what that means is we looked at patients who had original diagnosis of non-dysplastic Barrett's, and we looked at their outcomes 1 year out. So 1 year after their diagnosis, up to 5 years of follow-up, how many of these patients truly progressed to high-grade dysplasia and cancer? So the baseline incident progression was all patients with non-dysplastic Barrett's, indefinite for dysplasia, as well as low-grade dysplasia. So this was a cohort of all dysplastic as well as non-dysplastic. Now the test is indicated for non-dysplastic, indefinite, and low-grade, not for high-grade or EOC, because those are the patients, our guidelines as well as the data is already pretty strong and convincing. We should be treating those. We're not risk stratifying them. We're not surveying them. We're treating those patients. But patients who we need to risk stratify were these initial patients. So looking at the GAP1 study, it showed the hazards ratio of 9.4. So patients who had a high-risk TissueCypher score were 9 times more likely to progress to high-grade dysplasia and cancer when they were looked at 1 year out from their incident diagnosis. Now, looking at GAP2, this was a second validation study, and this is even more meaningful to me because this is the one that gives us sleepless nights. When we looked at patients who progressed to high-grade dysplasia and cancer, we found up to 25% of these patients originally had a diagnosis of non-dysplastic Barrett's within 1 year of their prior endoscopy. So that means these are the patients with prevalent disease. GAP1 looked at patients who progressed with incident disease. However, there are patients, despite doing the most careful exam on the most particular day, where we've done everything we can with Seattle protocol biopsies and adjunct brush biopsy tools, there is still patients we're going to miss prevalent disease. And this is what GAP2 looked at, is patients who had expert GI pathology biopsy confirmed non-dysplastic, indefinite, and low-grade dysplasia. And if they had prevalent disease, looked at patients from day 0 up to year 1, and looking at TissueCypher high-risk score, 46 times more likely to have prevalent high-grade dysplasia or esophageal adenocarcinoma compared to low risk score. So it demonstrates the ability of TissueCypher test to detect field defects that precede morphological changes. So defects that are there in plain sight, but we can't see them yet and who are going to progress. So the key validation part was validating the tool's effectiveness and these risk stratification. And it showed a really good negative predictive value, helping clinicians identify the low risk individual. So there is definitely the power of the positive, but there is a significant value of the negative. And it helps us identify patients who we can be assured do not need to come in for more frequent endoscopies, who do not need endoscopic ablative therapy, that these are the patients truly who are going to be not going to be progressing to something worse down the road. The other capabilities is finding the prevalent dysplasia. These are the patients that I worry about, that I never want to be that physician who did an endoscopy and within a year patient got cancer despite my best efforts. And this is where it helps identify the high risk, the value of the high risk score, suggesting the potential of misdysplasia. So the sensitivity is something that we have challenges in interpretation. And one of the things that comes up with this is, what about the sensitivity of 60%? Is there some skepticism remaining because it's not 100%? Now, when we talk about sensitivity and specificity, that's more for a diagnostic test. TissueCypher is a prognostic test, not a diagnostic test. So currently, our prognostication values are 0%. There is no test outside of TissueCypher that's going to prognosticate who's going to go to cancer or develop high-grade dysplasia. What TissueCypher does is it brings us from 0% to 60%. So in my mind, that's a huge ground that we gained. And that's why that's truly that value. So the 63% sensitivity calculation noted on the Davison study, it pairs the high and intermediate risk as actionable. And because there is debate among clinicians how to best handle this category. In my mind, if you look at the curve where it takes off is in that intermediate risk. So clumping the two together, both of these are actionable. And in fact, all three of these groups are actionable because there is the value of the low risk score, but the intermediate risk and high risk both have value in showcasing that increased risk. The other question comes about repeat testing. As we showed on the initial validation study, this was a 5-year risk prediction model. So patients can be retested at the next guideline-based surveillance endoscopy in the event of a clinical or new endoscopic findings. So if we find that the patient's Barrett segment length has changed, or symptomatically they have changed, or you see that there is some nodularity or something that's progressed, and despite EMR everything comes non-dysplastic, this definitely helps us risk stratify the patient to make sure that the risk score has not changed. The responsibility comes that the test should only be ordered by providers that can change or facilitate change in management. And in some cases, this could be our care teams, our advanced practice providers, or if we have fellows or trainees who are involved in the care, there is definitely value in having them make those decisions and then how to act on it. We don't want to order a test that we don't want to know what to do with the results. The patient cost, in our experience, Castle Biosciences, who is testing and delivering this test, has worked on behalf of the patients to submit, manage, and as necessary, create appeals with the insurance plan. And that is because the quality of care should not depend on the financial considerations. So there is a patient assistance program that makes that possible as well. So not only is there a patient benefit, but there's also the societal benefit of reducing the incidence and the morbidity of esophageal cancer. And that is truly making a societal impact. And then standardization of results-based practice of looking at endoscopic eradicative therapy for high risk versus extending the interval for low risk, ultimately reducing the healthcare expenditure. And Castle has been great to work with that, where it does not cause any additional financial strain to the patient or to the practice. So with that, I'll hand over the podium to Jen, who's going to go over some real world case scenarios and how we all incorporate this for the patient's decision making.
Thank you. It's an honor to be here, to be part of this group, and really just to work on developing this over the past few months. The goal is really to provide a pathway for how to use this in clinical practice. And so how do we take our patient in front of us and make this an actionable test? So we'll take the first case study, a 55-year-old male, chronic GERD, 20 years, obese, history of tobacco use. And you do your endoscopy and you find C0M3. So low-grade dysplasia Barrett's esophagus segment in a 55-year-old, you have a few options. Endoscopic eradication therapy for low-grade dysplasia, somebody of this age who may have want to get rid of the Barrett's entirely, versus a surveillance that would be pretty frequent surveillance to keep an eye on this. And I think many would probably opt for some sort of an endoscopic eradication therapy, but you'd want to sort of understand his risk and his preferences. So it's a great case for TissueCypher low grade. And you do the test, and it comes back as low risk. So when you look at the way the score is presented, you're going to get a few different pieces of helpful information. So the risk class is low, the score is 2.0, and importantly, the 5-year probability of progression is 0.8%. So on histology, in a 3-centimeter segment, this was non-dysplastic. This was low grade. But your probability of progression is extremely low, more in the realm of what we think of for a non-dysplastic. And so after a discussion with the patient, and of course incorporating their wishes, their preferences, this is actually a reassuring test. This is a case where you can downstage the risk. TissueCypher helps provide that reassurance. And you could have a few different outcomes here. Instead of proceeding with endoscopic eradication therapy, you could go to surveillance alone. And within the realm of surveillance, still within the guidelines, your options could be 6 months, 12 months. And I think in this case, it would be extremely reasonable to do surveillance at 1 year. So a clear scenario where TissueCypher was able to help downstage and reassure with an extremely low risk. Our second case study is a 63-year-old female, chronic GERD for 15 years, average weight, non-smoker. GERD is well controlled with her daily PPI. But now you have a very concerning family history, and this would probably scare many of us and probably scares the patient quite a bit. So she has a father who had esophageal cancer around the age that she is. Her uncle has a long segment Barrett's, and she has a short segment when you do your endoscopy. So she has C1M1, non-dysplastic. So endoscopically, this looks like a low-risk situation. But she's very worried, and her family history is also a bit concerning. So again, a really useful case where you would want additional information, and you really want personalized information. What is her risk based on her family, her genetics, her biologic profile? And so in this case, you send your TissueCypher, and it comes back as high risk. This is a risk score of 8, and the 5-year probability of progression is 28%. This is worrisome. And so you're going to have a clinical discussion. Not only is she high risk for progression with this strong family history. So the first thing that you might consider is, was something missed on that first exam? So you'd want to, I would suggest probably looking sooner because something may have been missed, prevalent dysplasia. And then I think this could certainly push you into upfront endoscopic eradication therapy, given this extremely high risk of progression for the C1M1. So two sort of very different cases that illustrate the value for patients. So you're getting a personalized, objective risk assessment. And when you have a high-risk test, this is actionable. This is going to streamline you right towards something that's more aggressive, which could be a more aggressive, more frequent surveillance, or going straight to endoscopic eradication therapy. The low risk is reassuring. There's value in this as well. So it reduces anxiety for your patient and anxiety for you as the physician who is more comfortable and confident in this patient's that you could say, I'll see you in a few years. It avoids overtreatment, and you can certainly lengthen the surveillance intervals. So it's clearly valuable for patients, for providers, and there's just some considerations about how to sort of make this work in your practice. And I don't think there's a one-size-fits-all, but it's definitely a team approach, and this is just something that you would want to incorporate, that your staff, your pathologists, the referring physicians, the physicians who may be doing the endoscopic eradication therapy, that everybody involved really understands the value and sort of what that TissueCypher score means, what that percentage progression means, and have some sort of an algorithmic approach. So with that, I'll hand this back over to Dr. Komanduri for the final key recommendations.
Thank you. So I think what you heard is a very thorough discussion so far around how to really look at this from the lens of your practice and practically implement this novel innovation to have some impact. And I think you can definitely see the value proposition. In my opinion, we're in an era of medicine where objective data drives all. We've spent way too much time in the world of Barrett's esophagus hypothesizing based on a very poor algorithm where every step of our process is fraught with error. So this is a very refreshing moment for us to have something that's data-driven, that also provides us with something where a patient can really visualize and understand, and both on the positive and negative side. I think our patient selection in terms of both high risk and intermediate risk was discussed, and really ensuring what was mentioned many times is that you don't take on a test for any risk stratification in any disease in medicine unless you're willing to take action. And if that's something that you can do or offer the patient, this becomes invaluable. I think a lot was said about workflows. As Jen highlighted, this is not a one-size-fits-all there. But that being said, this is not prohibitory. We've now most recently launched this across our entire healthcare network of 11 hospitals of various types in Northwestern. And really having a pathologist help lead this is very crucial. It can be very automated. I think the Geisinger group has shown that. And there are AI tools that can help this become even more automated over time. And ultimately, like we said, this is meant to help standardize some of the ways we manage this disease. I think we have a fantastic sort of opportunity in the next 5 years. We're not only outstanding at providing endoscopic eradication, but we're starting to screen for Barrett's. We're starting to screen for esophageal cancer. We're going to end up with a much bigger pie, a much bigger denominator of patients with non-dysplastic disease. And the old way of just looking at clinical factors and just sort of randomly making decisions on things which just don't have the risk prediction, as was highlighted here in the data, is not the way to do this moving forward. I think that's a small piece of it, and that triggers something objective like TissueCypher. And ultimately, I can't tell you how much patients appreciate, some of these patients had Barrett's for many years, and having a doctor come to them and say, hey, we're going to make this very objective and data-driven. We're not going to just guess and hypothesize and tell you that there's no data, because we have some. We have the ability to objectively risk stratify these patients and ultimately provide them with an actionable outcome.
Thanks, everybody, for staying on, and thanks to the panel as well, my colleagues, and Sri for this whole endeavor. And it's been great. Thank you, everybody.
Thank you.
Thanks.